Let's talk about proteins. The function within the cell of proteins is anything you can imagine. They function in energy storage. There are calories in proteins. Structure. They also function in moving the cell through its medium, moving things past the cell, moving things from the outside of the cell to the inside of the cell, outside of the cell, transport within the cell, enzymatic functions, they function in protection, you name it, a protein can do it. So let's talk about the structure of proteins. Proteins are composed of monomers that are called amino acids. Carbohydrates, remember, their monomers are monosaccharides. The monomers of proteins are amino acids. And here is the structure of your basic amino acid. We're not going to worry about chirality, so don't worry about the right hand, left hand. But you have a carbon that's attached to a side chain, and these can vary, but it has a hydrogen, an amine group, and a carboxyl group or an acid group. That's why they're called amino acids. Get it? Now, when we hook amino acids together, we form peptides. If you get enough peptides that are long enough, we call it a protein. So let's look at how that happens. Here's our amino acid, here's our carbon in the center, here's our side chain. In this case, it's a very simple side chain, it's just a hydrogen. We have our amine group over here, we have our acid group. Over here we have another amino acid, here's our amine group, our carbon with our hydrogen. Here the side chain is a little more complicated, it's a methyl group, and we have our carboxyl or our acidic group. So we remove the hydroxyl group from this side, we remove the hydrogen group from this side, removing water. This is becoming a theme, huh? Dehydration is generally how the cell puts all of its molecules together. And when you break it apart, it's called hydrolysis. Okay. So when you hook these two together through dehydration synthesis, you get what's called a peptide bond. And you start getting a string of amino acids. Now once you get past about 6 amino acids, and once it's functional, we start calling it a protein. If it's less than 6 or 6, we call it a peptide. When you get a string of amino acids hooked together by peptide bonds, we call this the primary structure. But proteins generally don't stay in this long, straight string. They start to fold. And the first set of folds we call the secondary structure. Now, there are two main types of secondary structure that we've identified that is important to the functioning of the protein. The first one is if they form a coil, going around and around and around and around. Ignore the purple balls. I'm not sure why they decided to put those in there. But this makes the molecule springy. It gives it an elastic nature, just like a spring. The molecules in your hair and in wool have lots of these helixes, so it makes it springy, elastic, so your hair can move around. Now, if you want the protein to be very rigid, very stiff, then you can form a pleated sheet. That's where the primary structure goes back and forth, makes a U-turn, comes back and forth, and we're forming hydrogen bonds between the different pleats. Proteins that are very stiff, that are very rigid, have lots of beta pleated sheets. Now, over here with the helix, one thing I forgot to mention, they're stabilized by hydrogen bonds. Now, hydrogen bonds are easier to break than covalent bonds. So if I pull on this side and keep this side stable, these hydrogen bonds break, they stretch out, I let go of it, bing! Back there it's forming those hydrogen bonds again, and that's where we get our elasticity. Whereas here, if I were to pull it on this side, it doesn't have as far to go. This U-turn here is going to keep it from pulling too far, so it makes the protein very rigid. Now let's move on to further folding. Now that we've done the secondary structure, the protein can fold further into more shapes, and we call this the tertiary or 3D structure. Now if you look at this molecule here, you can see there's an alpha helix there, there's a beta pleated sheet to keep it stable there, and then we have the rest of this, and it's being held together in this form. Now, remember that your alpha helix and your beta pleated sheet are held together by hydrogen bonds. And if you look over here, the tertiary structure has some more going on with it. Now here it's the side chains that are coming into play. So we have a side chain here that we've got an acid and we've got a hydrogen bond or a hydroxyl group, and they can hydrogen bond between the double bonded oxygen and the hydroxyl group. You can form a hydrogen bond and it's going to stabilize that part of the molecule. Over here, if you look at these side chains, this one right here and this one right here, there's no oxygen, no nitrogen. So can't hydrogen bond, no ionic interactions, so it's hydrophobic interactions. They're being excluded by those that can hydrogen bond. Now down here, you'll notice that I've got an amine group on the end of this side chain. It's picked up an extra hydrogen, so it's basic. It's got a positive charge. I've got an acid group that's lost its hydrogen, so it's negative. And this positive charge and this negative charge are going to be attracted to each other, and we're going to have an ionic bond. Then here we have a sulfhydryl group here. We've removed the hydrogens and formed a covalent bond between these two portions of the protein, further stabilizing it. So those are the four interactions that can hold together the tertiary structure: hydrogen bonding, hydrophobic interactions, disulfide bridges, and ionic bonding. Now sometimes you can take two different strings, two different primary structures, and put them together in what we call a quaternary structure. So you'll notice that we have two subunits for this protein. We've got this subunit over here, we've got this subunit over here. In this example, they're the same subunit, but the protein isn't functional until the two subunits come together. Now an example of this would be your antibodies. They're composed of 4 subunits that are bound by disulfide bridges. Collagen is another one. You get 3 molecules of collagen coming together to form the actual collagen. Okay, and we find that between the joints of our bones. This is called the quaternary structure. Now what holds these two subunits together are the same thing that holds the tertiary structure together: hydrogen bonding between the side chains, hydrophobic interactions, disulfide bridge, and ionic bonds. Now with all of this folding, you can make any shape you want, which leads to the vast array of functions that proteins perform in the cell. We'll be talking more about proteins as we go along. All of this lovely organization can be unraveled by breaking these bonds, the hydrogen bonds, the ionic bonds, and if you really work at it, you can break these disulfide bonds. And this denatures the protein or unwinds it into its primary structure, just the string. Now what happens when you denature a protein is that when you remove whatever it was that was breaking those bonds that held it together in its tertiary, quaternary, or secondary structures, then when it comes back together it forms a different shape. And generally speaking, it doesn't perform the same function that it did before. Think of frying an egg. When you crack the egg into your pan, the yolk is yellow, and it's cloudy, but it's a nice deep yellow, almost an orange, and the whites are actually clear and runny. The yolk is also runny, but once you heat it up, and heat will break hydrogen bonds, it'll break ionic bonds, it'll break disulfide bonds, because heat is energy and it makes this molecule vibrate. And as it vibrates, these parts of the molecule move further and further apart, and you denature the protein. And the yolk becomes a lighter yellow and it becomes solid, and the white goes from clear to white and becomes solid also. That's because the proteins in the egg are being denatured. Now, can you make that cooked egg become runny and raw again? No, you can't. Generally, when you denature a protein, it's non-reversible. When you denature proteins by heat or through adding extra acids, you can also cook fish by adding some lemon or lime juice, and that breaks these ionic bonds, denatures the protein, also messes with hydrogen bonding. Then you also cook it through use of an acid. Then you can't go back to the original function, and it kills microbes. It'll kill us. If you give a human too much heat, it'll denature our proteins and we'll die too, because the proteins denature and they stop performing their functions. Now, you can also denature proteins with things like Clorox bleach, but you wouldn't want to eat it afterwards because it would denature the lining of your throat and your stomach all the way down. Generally, we pick denaturing agents that come and go or don't bother us too much. Now, we're going to be talking about denaturing throughout the course of, well, this class, and I'll be bringing this up again.